Susie,
Rob,
what is the worst haircut you think you've ever had?
I think when I was about seven, my mum insisted on giving me one of those bowl haircuts. She got it done at hairdressers. It wasn't actually put on, it wasn't a bowl put on my head and her cutting around it, but it looked very much like that had occurred. It was terrible, a terrible, terrible. And obviously, I was seven and it was 80, so I also wore terrible clothes. It was terrible. How about
I
you?
thought that the whole point of a bowl haircut was that it was at home. It seems weird to go to a salon and get a bowl cut.
What's yours?
I don't think I think maybe just when I was I don't know 13 and my hair just ran wild. Like it was all down in my eyes and I looked like some sort of wild creature. But in terms of the worst haircut, I think the worst haircut might have been when I was during the pandemic and I tried to do it myself and slipped.
Oh
And then I had
no.
an enormous, like, the little attachment came off. So I got like a number zero for like a streak up the top of my head. And then had to ask my daughter to come and sort it out. So basically just had to shave my head.
Wow.
So the reason I'm asking you about the mad haircuts is because you're going to tell me about some mad haircuts in fossils, right?
That is right. We have a great fossil today to talk about, which is we've been nicknaming the Triassic weirdo, and it does have a fairly crazy haircut.
Well, I look forward to that. So, Susie, what have you been up to this week?
It's the summer holidays, isn't it? I've
Yeah,
actually been in France. I just got back from France on Friday, in the French Alps, mountain biking, hiking, that sort of thing. How
that
about
sounds
you?
good.
It's fun.
I've been staying in rainy Manchester. I have children downstairs. I should just point out they're my children, so I'm not keeping some weird children down there. So I may be interrupted for questions about snacks and Netflix at any point during this record today. That
Excellent.
that's the level of excitement going around here. Why don't you tell me about this week's paper then? What should we? What's the title? I've forgotten already.
Yeah, the title is Triassic diapsid shows early diversification of skin appendages in reptiles. And it is by Stephan Spiekman, Rainer Schock, and their colleagues. And Stefan and Rainer are at the State Museum of Natural History in Stuttgart in Germany. And there's a
Hmm.
whole host of other authors on this paper as well who did various parts of it. Now, the last paper that I did, Rob, was the one on pterosaurs and largurpetids. And I had to do a bit of a kind of conflict of interest statement at the beginning, where
Yeah.
I said that, lots of the authors were, in fact, our friends and my friends and stuff. And I have to do that with this one as well. I'm not just going to do papers by people that I work with, but I have to actually say that Stefan, the lead author of this study, was my former postdoc, and he's great. I love him. He's brilliant. And I'm very excited about this paper actually because I heard about these fossils quite a long time ago when Stefan was first offered this position to work on them, and he
Mhm.
told me in secret about them. And so it's very cool to see them finally see the light of day in Nature. So yeah,
It
this
is
paper was
cool.
published in Nature. Yeah, and just last week, so 23rd of July, it was published,
Mhm.
2025.
So this achieved quite a bit of press as well with some fairly mad reconstructions. This is kind of fun because we always like it when fossils look weird, but this one looks really weird in a way that's evolutionary interesting. This isn't just weird, it's evolutionary. Anyway, I forgot we were meant to start with some paleo fossil files updates, weren't we?
Oh yeah,
Um,
well, do
yeah.
do your updates. Do your
Yeah, so
updates.
thanks to the people at Paleocast, our friends at Paleocast have gave us a shout out, and also thanks to the other people at other paleo podcasts that we've been talking to, Terrible Lizards, and the other one who I've forgotten. Well, thanks to them for their support. We
Good
are
ad though,
close.
Rob.
Great. I'm sure they'll appreciate the shout out. So thanks to them, but also an update on our de-extinction episode before we go into the Triassic weirdness. There have been some. Okay, so update from our de-extinction episode. So I can't remember how many episodes ago we did an episode on the de-extinction of direwolves as purported by Colossal Biosciences, and we took our own cynical look at that. Well, since then, friend of the fossil files, Tori Herridge, and a number of other people have, I didn't realize this, had been subject to some fairly weird, coordinated, and unsubstantiated attacks, impugning their level of academic qualifications. And these people are the critics, the most vocal critics of the extinction, us not included. Apparently, we're not high profile enough to get these attacks. Have been facing some attempt to discredit them, and the attempts to discredit them are completely weak. Anyway, that now Colossal Biosciences have said they've got no knowledge of this and know nothing about it. But that I thought that was a bit of paleo drama for
That
us
is a
to
bit of paleo drama. So what are they how are they being discredited in what venues?
Well,
And what are people saying?
blogs, YouTube, a lot of it's anonymous. And when people are actually trying to ask the questions of who wrote this and who for, then there's like, oh, we don't know. And then some of them have got withdrawn because of that. But some of the
Oh,
criticism is saying, well, Tori is unqualified, which is bonkers. How could she be not be more qualified? She's an archaeologist who works on mammoths, has written articles about de-extinction, including the ethics. And some of these other archaeologists have been called into question. Again, it's not an actual substantiated, let's deal with the arguments about what they're saying. It's, oh, I don't think you're allowed to say that.
it's ad hominem attacks on some of our friends and colleagues. That's not
Absolutely.
very nice, is it?
Well qualified, highly ethical
Hmm.
people who have dared to criticize this. Now, colossal bias, just caveat before Fossil Files gets closed down,
Sued.
sued, because um, I'm joking about that because I don't think that they're going to even ever find
Listen,
out about it. But
no,
but the point being that I thought that was kind of shady, and there's no direct link between these pieces and the authors.
no. We have to be careful also, Rob, about saying, "Oh, it's okay. We can say whatever we want because people don't listen." Because this morning, when actually you released the pterosaur lagoderpatid one,
Yeah,
Davide Foffa, who was the lead author of that study, texted
yeah.
me and said, "Actually, I do listen."
All right. Okay. So we now need to be careful about this.
So people listen to our podcast, Rob.
People do listen,
Not that
and we
many, but
now.
some people listen, which
Does
is
that mean
nice.
Does that mean we need to watch our mouths?
Oh, I don't think so. No,
No.
no,
Okay. Well,
no,
with that
I
in
don't think so.
mind, not watching our mouths, let's go back to this Triassic punk.
Let's go back to this Triassic weirdo, this Triassic pump. Okay. So, yes, this paper. Um, okay, let's start. Um, Feathers in birds, of course, we're all familiar with, and hair in mammals. Very important key evolutionary innovations. Critical for things like insulation, so keeping us warm in if we're mammals, display, and also, of course, in birds, flight as well. But actually,
Hmm.
these two, hair and feathers, are just kind of two examples of integumentary structures, which
Hmm.
are, structures that come out of the skin. So integument is your skin. So an integumentary structure is some structure that comes out of your skin. And lots of different integumentary structures are being displayed by amniotes. So some others, for example, are scales,
Yeah.
claws, horns, things like that. And although they're all quite different and possessed by different groups, it's thought that the genes that allow amniotes to make these sorts of things, so the genetic toolkit underlying integumentary structures, was actually in place by the Carboniferous period, so about 350 million years ago. So
Hmm.
it's got kind of super deep evolutionary origins. So although we think that that toolkit must have been in place and those genes must have been there already by that time, there's hardly any evidence in the fossil record of integumentary structures in amniotes outside of the lineage to birds.
So, integumentary structures, feathers, scales, hair, that sort of
Things like
thing.
that,
Right. Yeah. Yeah.
deep evolutionary origins. And of course, in the fossil record, we really only have lots of evidence for these sorts of structures on the line to birds, where
Mhm.
we've got things like pterosaurs, which have these structures called, well, they're called pycnofibers, aren't they? Do you remember these? They're sort of
Yeah. I call
slightly
them fluff.
hairy
I call them
fluff. Yeah,
fluffiness. Yeah. Let's call it fluff.
pterosaur fluff. And then, of course, theropod dinosaurs and birds. There are actually some ornithischian dinosaurs, some bird-hipped dinosaurs that have some structures that seem to be some sort of integumentary structure, but there's a lot of debate on the bird line about whether these features are homologous and whether they're sort of feathers or
Hmm.
proto feathers, or whether they're actually evolved independently and different people think different things. And then there's the mammal lineage, of course. In early mammals, we also start to see fluff and eventually, of course, hair. And in fact, outside of these two lineages, there's only evidence for any sort of complex integumentary structure, big kind of display structure, is in an enigmatic fossil called Longisquama, which is from the
Hmm.
Triassic of Kyrgyzstan. Are you familiar with Longisquama, Rob?
Longest grammar is weird, right? That's. I mean, before I was reading this paper, I was aware of it because it had these. It looks like an absolute mad case. It's also interesting because of case study about how far can you stretch interpretations of these things. I guess the difficulty has always been with, as far as I understand, with longest grammar, is that as far as I'm aware, it's preserved as impressions rather than actual tissue, right? So to try and figure out what they are, is hard. So we've kind of got this question mark over this absolutely mad looking thing. I guess we should describe what it is. Some sort of reptile looking thing with enormous projections coming out of its back, far longer than its body, about five or six of them, but really long and thin. And I'm saying the word projections because that's hedging my bets as to what these things are.
Yeah, totally. And actually, as I usually do, and as I've said before, I did what all good paleontologists do and go to Wikipedia to take a look at,
Uh-huh.
what the fossil actually looks like. And they do have a nice picture of it on in Wikipedia, actually. Um, and I think part of the problem is that actually the front of the animal, I mean, actually in the fossil, it looks like it's drawn on,
Yeah.
but it's like there's hardly any of the actual skeleton preserved. It's just kind of the front half, and it's very squashed, as you said. And then it's got these long kind of structures coming out, and apparently they've been hugely debated, and some people even thought and at various times have interpreted them to actually be bits of plant. And so actually you've just got
Yeah.
like a dead reptile lying on some bits of plant, and people like that these things aren't actually belonging to this animal at all. um
So before we go further, thinking about, as all good paleontologists would do, they go to their Wikipedia page and look it up.
mhm.
But actually, if you Google some of these things, some of the first few links you go to might be slightly controversial ones, to put it mildly, in that David Peters of Reptile Evolution or Pterosaur Heresies website, depending on what you call it, he has, so he is, how would you describe him? Is independent, should we say?
An independent person.
Independent person who writes this website where he studies fossils and comes up with his own ideas about what's going on. And
He's
he
described
has
he's described as a paleo artist here. Okay, so
paleo
he's an
artists.
independent paleo artist.
His search engine optimization game is on point.
It
I
really
don't know
is,
how
isn't
he
it?
does it. Yeah, yeah. Whatever you Google, you'll come and find his page before you'll find something from someone sitting in an ivory tower. Uh, you'll come to this
You and
particular
me, for
conclusion.
example.
You and me, yeah, yeah. Well, that's because we don't write good enough websites, it seems.
Well,
Anyway,
clearly.
he has a pattern where he will take fossils, pictures of fossils. He doesn't study the fossils. He takes his published pictures of fossils and interprets what he thinks is going on through. He has this technique that I forgot what he calls, where he basically takes high contrast, oh, I've got to watch my mouth now because he has a technique where he makes the photos, edits them, and then puts on a lot of stuff. So Longest Squama looks weird, but then when Dave Peters looks at Longest Squama, he's added on a whole bunch of extra stuff, like wings and flying structures and whatnot.
I mean, he really has. It's bizarre, isn't it? It's
Uh,
yeah.
yeah,
It's You can go to the to if you Google David Peters reptile evolution and go to his, there is a Wikipedia page about him where he is described as being notable for his fringe views regarding
Fringe.
prehistoric animals.
Okay. Well, I think yeah,
Fringe
we
views.
probably shouldn't say much more about it beyond that. Maybe we do a dedicated episode on his opinions another time. But the longest one was interesting because that was often the one that first led me down this line of yeah. Let's take a
The
step back to
Peters
what you were
rabbit
saying.
hole.
Yeah,
Okay.
I've taken you on a rabbit hole. The point was there's this other weird thing from the Jurassic, and it's got this mad and how do we interpret it is actually really challenging. And you can go too far. Is it a plant? Is it part of the organism? That was where this context of this fossil, this new paper came from, right?
At this point, work on longest one was reached a bit of an impasse, hadn't it? Like some
Hmm.
people thought it was an archosaur, some people thought it was some sort of weird reptile, some people thought that these long structures were bits of plant. So but I think most people agreed that they were actually some sort of appendage, some sort of integumentary structure. But what the role of what the function and what the kind of significance of these long structures in Longa Squama were has unclear, and what this means for kind of the evolution of integument in amniotes was unknown. So let's move on to this new paper then, which in which the authors present some new fossils, and they name this new fossil as Mirasaura grauwvogeli. Now, it's known from a huge number of fossils, unlike Longosquama, which I think is just known from a few. It's known from around 80 specimens. Now, most of them are incomplete, but there are a couple of very nice ones which are much more complete. The specimens are 247 million years old, and they're from the Middle Triassic, or the earliest Middle Triassic, or possibly the latest early Triassic. So pretty early on in the Triassic, of some rocks called the Greater Voltsia Formation, which are in the Vosges Mountains of France. I had to actually ask ChatGPT how to pronounce the Vosges Mountains, because it actually is spelt Vosges Mountains. I was like, I bet that's not how the French pronounce it. And it turns
I was
out,
going to say, is it one of these parts of France that's actually German, but we're not allowed to say that because France would not like that.
well, let's not say that then, Rob, because you know what? We're already going to be sued by Colossal. We don't need to be sued by the state of France as well.
Macron's coming for us.
Macron's listening. He's coming for us. Okay, anyway, crashing on. The Grey Avoltia, which I'm probably pronouncing wrong, these rocks in which this thing is found, is actually a Largostata. It's famous Largostata. Largostatas are sites of exceptional fossil preservation. And in this case, it's very famous for its plant fossils, which are preserved as carbon films. And this is where this little specimen is from, or this little reptile is from. Did you see why they called it Mirasaura? Did you see what its name means?
No, I
Mirasaura.
didn't.
It means marvelous reptile. What a great name,
Oh,
marvelous reptile. I think that's brilliant. I think marvelous is a word I'm going to use more in everyday conversation.
yeah, I think that's fine. Oh, can I give you a little context? I mean, I don't know if you were going there about the species name. I did look, I didn't see
Yeah,
the genus name, but the species name. They've named it after the guy that found it, I think.
they have. That's right. Yeah. He found all the fossils. Yeah.
Grauvoglii. Now, when I
Yeah.
was reading the
Grab
news and
Vogel.
views for this one, they gave a bit of history of the specimen. Were you going to
Oh,
come
did
to,
they?
or am
No.
I,
Yeah. Why
no,
don't?
no.
Yeah. Yeah. Do
Yeah, so
enlighten
this
me.
guy, this Voglier, Grauvoglier, he collected it in the 1930s and it's been sat in a private collection for 70 plus years. Uh, and people knew about this specimen and it was just sat there. Now, the reason I mention it is because this loops into our conversation we were having a few weeks ago about private specimens, right? So this amazing specimen was sat in these collections, but of course it's just gathering dust, but if it's in someone's private collection. But when it eventually, I was going to say, was donated, transferred, acquired, I don't know, by the Stuttgart Museum, then now people can work on it. That's when it suddenly becomes part of the realm of science. I thought that was an interesting observation.
That is, and another interesting observation that also circles back to previous discussions we've had around including authors who are non-academics who are involved with the collection of specimens and things like that.
Hmm?
Leah Grauvogel Stam is presumably related. I don't. Did I
Oh
read that she
yeah.
was a daughter, or I forget. We'll have to go back and have a look at what it is. But she's actually an author on this paper, and
Oh
so
yeah.
she's clearly part of the Grauvogel family who originally found this material. So that's quite nice, isn't it?
That's cool.
To include some of the people who originally, or the family of the people who originally found it.
Hmm.
Anyway, marvelous reptile. So Mirasaura. It's actually tiny. Did you see the scale bar? So we've got some lovely figures. Figure one of this paper shows some nice fossils of this specimen, and they're actually tiny. So the holotype is only about 5 or 6 centimeters long. So it would literally fit in
Wow.
the palm of your hands. It's just a
I
little tiny
completely
thing.
missed
Now
that.
they do say, unless I've completely misinterpreted the size of the scale bar, if I got had a horrible centimeter, millimeter. No, I'm sure I haven't. The scale bar is 20 millimeters in A. So there you go, 2 centimeters. So it's a little tiny thing, 5 or 6 centimeters,
Yeah.
sit in the palm of your hands.
Yeah.
Cute little thing. But they do say somewhere in the paper that actually, I think based on its brain case, it's got and structures in its skull, they can tell that it was probably quite a young juvenile, the holotype, and there are bigger individuals, but not much bigger. So they probably, I mean, an adult might have been 10, 15 centimeters or something. So little
Hmm.
tiny things, really. And of course, what makes it marvelous is that it has this series of around 20 elongate feather-like structures extending from its back straight up. And they're not paired, they're in a single line. This was something of a debate with Longosquama, going back to Longosquama, which also, of course, has these long feather-like structures sticking up from its back. Some people interpreted them to have been paired, but actually here in Mirasaura, they're not paired, they're clearly a single line. And they're
Hmm.
feather-like, really just in terms of their shape. So they expand as they extend upwards, they sort of expand out. So they're kind of wider at the top than they are at the bottom. And they've got a central spine, a bit like a
Hmm.
vein of a feather. And then either side of this central spine, there's kind of some sort of soft tissue, but but critically, when you look at that soft tissue closely, you don't see branching. So like in feathers, how you have the rat, I think they're called the ratchets, aren't they? The sort of
Hmm.
barbs that stick out from the side of the central vein. You don't have that in these. It's just a single kind of film of soft tissue that
Hmm.
extends down. So they only superficially resemble feathers. When we look at the structure of them, they're clearly quite different from
Hmm.
feathers themselves. So they're not that similar.
Yeah, that's pretty cool. I think also, I guess the important difference here is that the preservation is nice. We actually have
Yeah.
some, with Longus Guama, we've got these impressions, but here we've got some really fine detail and some films. So we've got some, as you mentioned, we've got some actual tissue, and that might allow us to do some more, informed understanding about what it is, how it was preserved, that sort of thing. Some of these
Yeah.
isolated ones in particular look really nice
They're
in
incredible, aren't
terms of
they? So
preservation.
there's quite a lot of extended data, as you can in nature papers. You can, just for the online version, you get this
Mm,
kind of extended data at the back, which allows you to put loads more pictures in than you
yeah.
otherwise would. And they've got some lovely pictures of all of these different kind of integumentary structures up close. There's lots of isolated ones, aren't there? I guess they're, because this site is best known for its preservation of plants. They were, I think when they were originally collected, I read that, people thought that a lot of them were plant fossils originally. But before we get more into the integumentary structures, Rob, I thought there are some other interesting things about Mirasaura, which actually help to solve a bit of a long-standing problem with Longus guama as well,
Uh-huh.
around the question of what is it. So it has, because we actually have a couple of full skeletons of this thing. The authors also put their fossils in the synchrotron. We talked about the synchrotron before. Very, very,
Hmm.
very kind of high resolution, super duper particle accelerator that allows you
Yeah.
to do very high resolution CT scanning. And Stephan, the lead author of this paper, is also a complete whiz at CT reconstructions. So he's done a beautiful CT reconstruction of the very fine details of the skull of
Hmm.
this animal. And what they know is that it has a large domed skull with forward-facing eye sockets, and the front of the snout is toothless, a little bit like a bird.
Hmm.
Um, Now, do you remember back to, learning vertebrate evolution as an undergrad? And, all of there were important things in the skulls, right? There are important
Are you going to
holes
start talking to me
in the
about
skull.
fenestra? Because
I
I'm going to
am.
tell you I have forgotten all my rules with my fenestra.
Do you not
How many
teach this
holes
stuff
do you
these
have in
days?
your head? I don't
Yeah.
know how many holes I have in my head. I should know. I mean, yeah,
Well,
you tell me.
I could enlighten you. So this thing has a supratemporal fenestra and an infratemporal fenestra. So it has two holes at the back of its skull. So the supratemporal fenestra is a hole that sits on the top of the skull, and the infratemporal fenestra is one that sits on the side of the skull. And these make it a diapsid. So diapsids are a group of reptiles, or they're a group of animals, that a group of amniotes that diverged from synapsids. So we've got the diapsids and the synapsids. The synapsids go on to be the mammals, and the diapsids go on to be the reptiles, everything that we kind of know as reptiles today, the reptiles, the crocs, the birds, and all those sorts of things. So first of all, we can be really confident that because of these two holes on the skull, this thing is a diapsid. However, it lacks an antorbital and a mandibular fenestra. So an antorbital fenestra is a
Oh
little hole in
God.
front of the eye. A mandibular fenestra is a little hole on the side of the jaw. And these are classic archosaur features. So
The
this
reason I'm saying
means,
"oh God" is because it's giving me flashbacks to trying to remember all these things and learn it all many years ago when
and
it felt like
label
this
it
was
in
really
exams.
important. It
Yeah.
doesn't feel important anymore, but it is important because it helps us know
Actually,
it is. Sorry, see, for the people at home that are listening, I want to say when I said it isn't important, Susie's face almost exploded with the idea that this is not of the most important thing.
that fenestrae, skull fenestrae, are not important. Well, let me tell you, Rob, that these skull fenestrae have allowed the authors to not to actually solve some long-standing issues in diapsid phylogeny here with this taxon.
Yeah,
So
yeah.
what they show is that it's clearly not an archosaur because it doesn't have these mandibular and antorbital
Right,
fenestrae.
right.
So this is important because people had previously suggested that Longus guama something which looks quite similar, at least superficially, to this animal, might have been an archosaur, not an archosaur. What are archosaurs? Archosaurs today are the animals which are include the birds and the crocodiles, but in the past, of course, they included all sorts of things like the dinosaurs and a whole line of extinct animals that led up to the crocs that we might call pseudotsuchians. Um,
So I got a couple of questions about that. I mean, I don't want to spend too long talking about fenestra and holes in the head, but you've sparked my curiosity now, especially because your indignation at how important holes in the head are. The this reminds me of that thing preserved in amber that was that
oh
small
yeah.
birdy lizardy thing. Because there was again, is it a bird? Is it a lizard? We should be able to tell the difference. But actually, there was an genetic effect that it was a small thing and it ended up looking a bit birdie when actually it was a lizard. And I don't know. This one now reminds me because it's quite small. But anyway,
Do you know,
that's not. Yeah.
that's actually exactly what I thought when I saw the skull of this thing. I was like, oh, it's like that thing that was interpreted as a bird. What was it called? God, I can't remember.
Oh, well, it
It was
doesn't matter
interpreted
anymore.
as a bird.
Okay, so just to give people a bit of a context, there was this thing that was held up as this bird preserved in amber. It's very, very important. Or was it a dinosaur? I can't remember.
No, it
And there
was
was much,
birds.
much hurrah, and there was question marks. So, well, first of all, how did you get a hold of this fossil? It's very unlikely you did this legally. So there's an awful lot of ethical issues around it. Second of all, you got it absolutely wrong. It's a lizard. Third of all, how dare you? So that was a bit of an embarrassment. That I think the name is now gone. It no longer exists. I think, unless they renamed the
Well,
lizard. I don't know.
well, they might. Well, I thought I would have thought that the name now is just applied to the lizard later, but
Well,
I
this is
don't know.
probably the time for me to admit that I didn't finish reading the entire paper. I'm relying on you to carry me on.
It's only 2 pages.
I know. I went out of time. It's the school holidays. I got up
I
to
know.
phylogenetic relationships, but I didn't get onto the. Well, I shouldn't explain all that.
Well, that's what I'm going to be talking about because what this thing has is not only does it have these missing holes in the side of its head, which show it's not an archosaur, but it also has a whole range of quite strange features that unite it with this very bizarre clade called the Trypanosauromorphs. And these are very weird Triassic reptiles previously known only from the late Triassic. And actually, when the authors have put Mirasaura, Longisquama, um, these two taxa into sort of general diapsid phylogeny to work out the evolutionary relationships of these animals, they found that Mirasaura and Longisquama are sister taxa. They're most closely related to each other. So they're very closely related animals,
Nice.
perhaps unsurprisingly, given what they look like. And they are both trypanosaurs. So these are trypanosaurs are these weird kind of probably arboreal. So they lived in trees, little tiny weird things. They've got some weird adaptations of the hands and feet
hmm
that suggest they might have been arboreal. They have these kind of pointy snouts with these forward-facing eyes, and they
hmm
have they sort of some adaptations for digging apparently in some of them as well. So very strange things. Not quite sure where they fit, but anyway, it seems that Mirasaura has helped to stabilize this and solve some of these questions around trypanosaurs and also Longisquama, of course. So now it's clear that they're non-archosauriform diapsids, so they're lying outside of the radiation which leads to birds and crocs, and also the sauria, the radiation which leads to the lizards and the squamates as well. So before that, some people thought trypanosaurs might have been some sort of archosauriforms, but clearly that's not the case. The authors find them to be much, much, much earlier diverging than that. So these nice fossils have helped solve kind of a little other conundrum about what these things might have been as well, and they're obviously trypanosaurs. Anyway, the main point about this is the integumentary structures.
Yeah.
So as we said, many of the 80 fossils of this thing, Mirasaura, actually preserve these integumentary structures. And so the authors did all sorts of things to these fossils, including, as we said, putting them in a synchrotron and also mapping the chemistry of them
hmm
to try and understand, what was the preservation of these preserved soft tissues. They're preserved as carbon films. It enabled them to visualize the structures much better, definitely show that there's none of those branching structures that we see in feathers. So similar to feathers superficially, but actually rather different when we look in detail. And also what you can see is that sometimes this kind of soft tissue down the side of the vein is actually folded and wrinkled in some way. So it probably indicates that this was some sort of pliable structure.
hmm So the big conclusion is that this is something weird and different, right? But mammals have got all hairy and fantastical. Birds have got their feathers. This is something else. This is this weird projection thing. Now, because I didn't read this part of the paper, I need to watch my mouth, I guess. But what about reptiles? Some reptiles have weird stuff going on, right? Iguanas have spikes going on the bottom of the top of their back, and some of them also have very fantastical gillar folds. That again, I'm
Yeah,
doing this
like
in a
folds
visual.
of
So,
skin, and
yeah,
they
yeah. Some
do. They
reptiles
have
have this
things
thing under
coming
the
out. Yeah,
chin. Yeah, they can
yeah.
do all these sorts of projections, and oh yeah, some of them with the
Oh
bearded
yeah,
dragon with the
yeah.
flared mane, I guess is the word I'm looking for. So there's an awful lot of weird, complex scale projections in um in modern reptiles.
Yeah, that's true.
Are we placing a big barrier between this and them?
Well, I guess it appears to be it's not skin-like. So I think a lot of
No.
those frills, those gular frills, again, I'm talking like I know something about this and I don't at all, but
Hmm.
they're projections of skin, aren't they? Those things, like
Yes,
that
yes,
thing
they
that
are.
flops up and shakes in that, yeah.
Yeah.
They're whereas these are clearly some other sort of tissue, some sort of proteinaceous,
Hmm.
keratinaceous maybe tissue or something like that. Elongations of scales, maybe
Hmm.
something like that, which I mean is what feathers are. So they don't really talk a great deal about exactly what this would have been made out of, probably because we don't know.
Hmm.
But what they do have when they put these things under scanning electron microscopes is melanosomes.
Yes.
So tell us about melanosomes, Rob.
Well, I mean, the fossil file fans won't need a reminder because they'll definitely remember everything we said about melanosomes from episode number one in the Pliosaur paper. Yeah, that's that.
Absolutely.
But just for those that didn't, that maybe are new. So melanosomes are tiny subcellular structures contained within cells that convey pigment. So they're inside your hair, inside your skin. They can be ball shaped or sausage shaped, but they're packed full of melanin and they give you color basically in your integument.
Yeah. And
Oh, and
what
actually,
they
also inside your body as well. Indeed, we mentioned in episode one, the team in Cork and the Cork or a lot of the Cork team are on this one too. Yeah.
they are, so they did the melanosomes work on this. And they what they did here was they put these structures under scanning electron microscope and found melanosomes in them. And of course, where melanosomes were first found in the fossil record was, of course, in bird feathers. So this is
Mm.
kind of significant because we've got these very feather-like, superficially feather-like structures, but they also have melanosomes in. So they were also obviously pigmented in some way. And we know also that melanosomes give kind of browns, greys, sort of subdued colors. You don't get like turquoise or yellow from a melanosome, right? Because those
No.
colors are produced by, their structural color. They're produced in different ways.
Yes.
But melanosomes give sort of browns, reds, grays, that kind of color to things. Um, and what the authors of this paper did was they actually measured the kind of aspect ratio of the melanosomes, so their length versus their width, and they did a PCA analysis. Now the results of this PCA analysis are shown in Figure 2. It's a graph at the bottom. How do you feel about this graph?
Well, this is a bit tricky because I didn't read it in detail, but there's a
Just
lot.
first impressions of the graph.
Yeah. So we've got a
Uh,
distribution. So this is basically the sausages and the balls. Sounds weird to talk about it in such a sense, especially when you put them together like that. But the meat and two veg of it, it's important because the shape of them is what can carry the different kinds of colors. And also the size of them is important. They mention the size in here because that's important about whether or not they're
yeah,
bacteria, whether or not they're more closely related to mammals or other things. So we've got this big distribution of bird ones. They're massive. Basically, all the space is taken over by birds. Birds have very long ones, very small ones, very wide ones, very narrow ones. And then reptiles have a smaller distribution. And then these fossil birds have a wide distribution. And this, what's it called again?
that's a
Mirasaura?
pterosaur. Yeah. Oh yeah, the purple thing's a pterosaur, by the way.
They're everywhere. So basically, everything's on top of everything, as far as I can
Right.
tell.
So
Yeah.
this is what I felt about this. I was like, well, everything's on top of everything. Now, from this, the authors suggest that actually Mirasaura's melanosomes are more similar to those of birds than they are to those of mammals. However, to me, they're all just lying on top of each other, and they're all kind of, birds just fill a very, very, very broad morphospace of melanosomes. The conclusion is that from this sort of work that they've done, that the melanosomes are actually more similar to birds than they are to mammals, melanosomes. Despite the similarity in the melanosomes and the apparent similarity of these structures to feathers, it is only apparent because these features, these are present in a very early diverging diapsid.
Hmm.
And because this similarity does appear to be completely superficial, so the main vein of these structures, the central kind of ridge of these structures in Mirasaura is completely different from
Hmm.
that of birds, and also these kind of pliable soft tissue down the side is completely different from birds.
Yeah.
So they're
I,
clearly independently derived.
I was going to say, I think it's very, they've made a strong case that it's not feathers. They've made a strong case that it's integument, and they made a strong case that there are melanosomes on it. And that's all part of the interesting story that is the main conclusion. And that is the main conclusion. Oh, and the phylogenies as well, which they used to convince us that it sits in this part of the tree, not another part of the tree. So hats off to whoever did the phylogenies. I like the phylogenies.
Yeah, they're
The,
good, aren't they?
yeah, so that means that lots of nice evolutionary trees where they've actually properly interrogated them and looked at alternatives and said, okay, if I force it to be in this part of the tree, is it better or worse? And actually, it's much worse. So
Much worse,
altogether,
yeah.
that's the main conclusions are completely, I'm convinced.
Good. I'm glad you're convinced. So what were these things for? Is I guess the final part of the paper.
Hmm.
Well, they're not, they're restricted to a single midline row on the back, projecting up. The authors say they think it's very unlikely that there was any sort of musculature
Hmm.
associated with them.
Hmm.
There's no clear muscle attachments, and you'd need quite big muscles if you were going to like lie them flat down on the back or pop them
Hmm.
up or something. So the conclusion is that they stuck up like this all the time. They must have been quite annoying. If you were carrying this around and you just
Yeah.
get tangled up in vegetation. I'm,
I,
I'd be surprised if they didn't have broken ones. Like, it'd be interesting to see whether there was any evidence of breakage of any of these things, or whether they could grow them
if,
back.
yeah, if I think about modern reptiles that do that, they have the ability to put it away. That gullah pouch that comes flying out, they tuck it away. If these things are sticking out of your back and they're double your body length and body height, and they're rigid and stuck like that, and you live in the trees, no, no, it seems like that seems ecologically implausible that you're walking around with them actually on your back.
But then
Um,
here it is. We have the
Here it
fossil of
is.
it.
And also, here's a question for you: if they were retractable, this is again another question about preservation and taphonomy. If they were retractable, when you died, would you be more likely to be died in a position of them being extended or retracted? I don't
Well, I
know.
mean, I guess that's the thing is that they've got 80 fossils of these
Yeah.
structures, and all of them are extended. They haven't got any that are lying flat. Although, would you
Hmm.
recognise them? Would you have recognised them as a feather, as the
Yeah.
same structures if they were lying flat? But also, do you think? I mean, they say that most of these 80 odd fossils, I think there's 76 or something of them, are actually just the integumentary structures without the body. So could they have shed them? Could they have
Ooh, that's an
grown
interesting
new
point.
ones?
Uh, Interesting. I don't know.
I've no idea.
Some of the pictures are of them just isolated. But the
Yeah.
question I was going to ask was when it comes to these trypanosaurs, which I've got to say I've never heard of before I read this paper, even though I'd heard of Longus this weird group of Triassic critters. When we think about the presence or absence of these things, how it's quite possible that these structures were much more widespread, but we don't know about it, right? All these other microsaurs, which were reconstructed as, sorry, not microsaurs, the trypanosaurs,
Trypanosaurs, yeah.
they're reconstructed as just something like some weird looking Triassic reptile. Maybe they were all covered in these things and we wouldn't know because you'd have to have a window of preservation that was really nice to say that it wasn't preserved. Because
Yeah,
you could
absolutely.
yeah, and they did that in the phylogenetic analysis. They went through a process of saying, well, how confident could we say it is absent? Because this is something that I sometimes think about my own research. If you're going to put in a zero saying this is definitely not present, you actually need to be quite confident about that. And especially with a soft integumentary structure like this, really, we've got a lot of question marks around. Are they there? We don't know.
Whereas the absence of evidence isn't the same as evidence of absence thing, right?
Absolutely. That's a far
Yeah.
more eloquent way of putting what I was trying to say. That's my
I
mantra
mean,
all the
if you just want
time.
to cut out all that stuff that you just said and just put in like that, that's fine. Just see how you feel about it when you're listening back to yourself, Rob.
Yeah. I see. If I leave it all in, I don't know if that reflects badly on me or you. Definitely not.
Anyway,
Okay.
so I mean, I guess the conclusion what in terms of what their function for is probably for display. I mean, that's where we go, isn't it?
Yeah, yeah,
Whenever we have a thing that we don't
yeah.
know what it is, we go, well, it must be for display. And
Yeah.
we're not. It's probably not wrong, right? It's got to be display. It doesn't have any obvious function. Wasn't for
No one,
flying. Wasn't for keeping warm.
no one's going to argue with that. There's all sorts of sexy structures that evolve all the time across living groups and fossil groups. But it's very difficult for a fossil to say this is a result of a sexual display structure, though, because you can never test that. It
No.
gets a bit hard. But I
Yeah.
mean, in all fairness, that is most likely the explanation for this. When you
Some
start
sort of
ruling out the
display.
other ones, yeah, this wouldn't have kept you warm. It wouldn't have been much use for anything, really.
No. It so what bearing? I mean, we started this with the kind of a discussion about the evolution of integument and this kind of genetic toolkit of integumentary structures in amniotes at the beginning. And I was trying to think, what bearing does this have on that? I mean, I guess what it does, it tells us that it's another example of a different integumentary structure that is neither feathers nor fur.
Yes.
It's complex integumentary structure, and it's occurring in a totally different group. So it's not in a synapsid, it's not in an archosaur, it's in a trypanosauromorphous basal diapsid lineage. So
Hmm.
it's another example of a type of integumentary structure that is
Yeah.
occurring on a group that's diverged fairly early.
I agree with you. That is the sort of evolutionary story. I think the other story is it's cool.
It's
That's
cool.
it.
It's
It's
cool and
cool.
it's weird. And that the Triassic,
Yeah.
it just kind of underscores this time that the Triassic was the time of weirdos. There was just
Yeah.
so many weird things in the Triassic. Now, the Triassic's really long. So I
Yeah.
will say this, it's like
Yeah.
it wasn't like 10 million years. It was super long time period, but it was full of just really strange reptiles. And here's another one.
It's a new weird data point, and it's weird.
And cool.
I'll just say weird
And
and
that's
cool
that.
over and over again. Yeah.
Okay. Yeah.
Yeah. So what's our take-home message? Well, you've already done the take-home message. Normally I like to
I
conclude
think
on the
I
take-home
think
message, but you just told us.
I think it is. I think that's the take-home message. I mean, weird, cool, and a new type of integumentary structure. So yeah.
Yeah. Um, Side note, this is, I don't know if I should say this now, but whilst at one point after you mentioned this place in France it was from, I had a little look at the map and the region because I was trying to figure out where these fossils are from and how widespread they are. Let
I
me
did
tell you
get the
some of the.
impression that you weren't listening to me. Glad you were just
I
Googling
was
while
just
we were
I paid
talking.
100% attention. Let me tell you some of the place names of the area where it's from. Wollerdingen, Ermendingen, Klauhausen, Walhambach,
So what you're saying is very
Montbronn.
French. Very, very
Yeah,
French.
yeah. Do those sound French to you? I don't. Now we're starting. I mean, Germany, France fighting over the Alsace has been somewhat of a source of contention. So maybe we shouldn't end on that bitter note. But I just wanted to point out that this is from, let's say Alsatian. Let's say it's from the Alsatian area.
And if I have pronounced Vosges mountains wrong, I apologize to all our listeners from the boundaries of the French and German Alsace region. And you can blame ChatGPT because that's
Yeah,
how it
yeah.
told me to pronounce it. That's not my fault.
Great. Well, in that case, I think that probably draws to a conclusion this Triassic punk. I think it's super
Yeah.
cool. Thanks for doing this paper. I mean, I guess we kind of had to because it's super cool. And well done to the team for that one. They've got a interesting discovery to their name. Also makes me wonder now how many things are left in collections. To pick up the private collections theme, how many things are just sat there in dusty drawers waiting to be put into a museum where someone can actually work on it and do some science?
Well, not just that. I mean, things that we need to go back and look at in our own museum collections and in collections that even that we can work on. There's so
Yeah.
many fossils, so few paleontologists, discoveries out there waiting to be made. Now, actually, also to increase the controversy, I think the longer Squama fossils are all in Russia, so currently inaccessible, of course, as well. So
Hmm.
difficult to go back and revise those and compare them and have a look again,
Well, they're not
because they're
inaccessible if you're Russian, I guess, but
they're
they're
not
inaccessible to
inaccessible if
us.
you're
Yeah.
Russian, but
Yeah.
for certainly for the authors of this paper, they would
Yeah.
have been. So yeah,
But I guess the other thing to point out is that this highlights that these integumentary structures, these mad bonkers things, are things that we should be looking out for. It's quite possible. This is the scene that we had with the plesiosaur one, right? You might have not been looking for it before, but now we know you should definitely be looking for structures that might be hard to see unless you've actually got a search image you're ready to go and look for now, which is, I guess, would accord with what David Peters would say. Now we can go. He and I are now in agreement. This is not where I expected
excellent.
this to
Well, at
go.
least that's one person who's not going to sue us today, Rob. Great.
I wouldn't guarantee it. I mean, well, so hang on. Who's now going to sue us? We've got the team from that
Colossal
worked on that
biosciences.
Colossal Biosciences, David Peters, the Myanmar Amber people that did that. I'm not going to name them because that would just encourage
No,
them to sue us.
the entire French state.
Oh yes, and Macron as well. Yes, yes. Okay. Thanks, Susie. I have to think about what I'm going to present to you next week. Maybe I'll do you some beehives.
Oh yeah, okay. All right,
So,
grand.
if we want to know about a massive paleontology fail,
That would be
maybe we'll
fun.
do
We could
that.
do a
Yeah.
massive for the end of the summer holidays, a massive paleontology fail.
Yes. Right. Thanks, Susie. See you.
See you.
The Fossil Files was presented by Robert Sansom and Susanna Maidment. You can find more information, including links to the original papers that were discussed, at fossils.libsyn.com, and you can contact us at fossilfiles.pod@gmail.com. Right, I may have to some of the libelous things I've said in there may need to go. I don't know about is the German France thing, is that just stoking trouble or is that
I
okay?
don't think it's staking trouble. I think since the decline of the Habsburg Empire, there's like we're probably safe.